Dr. You're going to talk about two methods today, right? One is trick or treat, the other is Lamin, right?
Yeah, that was the plan. But if you want to discuss more about one, that's also fine. So
no,
please
that's okay.
feel free to interrupt me to ask questions, and then we'll see.
Sure.
I can adapt. I can adjust. The content of the talk.
Got it. Any good news in what journals that you're going to right now?
Say it again. What journals?
Yeah, are we looking at for trick or treat? Is it already submitted to specific journals? Do we have any good news?
Yeah, it is submitted. So we're still hearing back. So we can let you know later because it's quite uncertain now. So
got it. Once we upload the video of today's session, we can actually just share the YouTube with editor of the journal that you submit to. Hey, you see, we have a great audience, sort of peer reviewed, by a bunch of sophisticated folks.
Okay.
So, okay. I don't know whether that helps, but anyway, I think I see people are still joining in. So I'm just gonna get started anyways, and give folks some time to join in. Hi, everybody. My name is Jiarui. I'm the volunteer lead of DaShu. So welcome joining on board today's session. This is the first talk that we're hosting this year, 2022. So, Happy New Year, everybody. Today, we're very fortunate to have Dr. Hongkai Ji from Johns Hopkins joining us today, giving the first talk of the year. I'm very excited. I hope everybody who signed up is also feeling very excited. And Dr. Ji came to our DaShu a few years ago, give another single cell topic talk, which is very successful. So we're happy to have you back, Dr. Ji, and welcome. Always love to have you back. So he's going to talk about statistical analysis of single cell RNA sequencing data. We're going to focus on multiple sample data analysis, some dynamic interaction of gene cells and samples. It's a very important talk to the community and to the field. That's why we're very much looking forward to hearing the specifics. So before I give a sort of formal introduction of Dr. Ji, while people are joining, so I just going to spend a few minutes very quickly to introduce DaShu to you. Many of you are new members to us. So first thing for all, you have our best wishes from our board team members and the volunteer teams. And we want to wish you guys an early happy Chinese New Year. Is going to come up in 10 days and a happy 2022. We're a very happy family. Feel free to join us. And Dr. Ji, if you're interested, become a board member, we also welcome you, given you're the old friend of ours. So what do we do? Well, we're a 501c nonprofit organization founded about 7 years ago. We're 7 years old. Before pandemic, we used to host a lot of scientific conferences, both virtually and on site. We have educational seminars, workshops, and we have career development workshops for everybody to come and social. But given pandemics, everybody is sort of working from home a lot. And for DaShu, we have sort of migrated a lot of activities virtually online. So we host our monthly seminar like the one you're joining today. So stay tuned for our very good topics and great talks, from industry, from academia. You're going to hear a lot from famous professors and scholars like Dr. Ji today. So stay tuned and feel free to, ask any questions if you're interested. And also every time if you sign up, our webinars, please donate. Your donations and support is very important for us to run the whole organizations. A few bucks would be very generous if we can have that. And also for many of the trainees, if you're graduate students, PhD and master students, feel free to reach out to us, and we're definitely want to have your voluntary support. If you want to become a volunteer, you can actually scan the QR code or reach out to all sorts of social media on LinkedIn, our WeChat channels, and leave a comment to us if you want to be a volunteer. We're a very happy family, and so your support is very significant and important to us. And all right, I don't want to take too much of time today because Dr. Ji is going to talk about a great topic focusing on most of cutting edge analysis of single cell data analysis. So Dr. Ji got his bachelor and master in engineering from Tsinghua many years ago, and he got his PhD and master in statistics from Harvard University. And then he joined Johns Hopkins, probably like more than a decade ago, 2007, right, Dr. G? And he's also an elected member of ASA, who had a chief in statistics and biosciences. If you search Dr. G's publications on PubMed, you're going to see a lot of very very much cutting edge analysis, method development on single cell data analysis. His research interest is very broad and big and in depth across all sorts of omics data, big data in performing machine learning models, hierarchical models, Bayesian methods, and a lot of computational, cutting edge analysis. So today, I think Dr. G is going to talk about two recent publications, very novel, fresh from his lab, focusing on analyzing single cell data. He's probably going to talk about tree-based correlation screening visualization first, and then we're going to look at some of the method development from the pseudo-time analysis. So without further ado, I'm just going to hand over to Dr. G. If you can start sharing your slide, that would be great. Thank you very much. Oh, by the way, we're going to have everybody muted during the talk, but if you have any questions, you can actually put your questions in the chat box. We're going to get to them in the last 5 or 10 minutes or so to address them. So I'm going to start sharing my slide, and Dr. G is all yours.
Okay. Can you see my slide now?
Yes, I can see your slide.
Okay, so it's in the full screen mode now, right?
It is.
Okay, that's great. And first, Jerry, thank you so much for your kind introduction. And it's my honor to be the speaker for the first seminar of this year. And I also want to thank Da Shu for inviting me. And for today, as Jerry said, I'm going to share some of our recent work on analyzing single-cell genomic data. And as you know, single-cell genomic technologies have been rapidly transforming the biomedical research. And in the past 10 years, you can see many single-cell genomic technologies have been developed. And unlike the conventional bulk technology that measure the average behavior of a cell population, single-cell genomic technologies measure the behavior of each individual cell. And take gene expression as an example. If you use the conventional bulk RNA-seq to analyze a biological sample, you would obtain a vector of gene expression values. Here, each entry corresponds to a gene. It measures the expression value of that gene averaged across all cells in that sample. Whereas if you use single-cell RNA-seq, what you're going to obtain is a matrix consisting of gene expression value for each gene in each cell. And with this data, you can now really study the behavior of each individual cell so that you can see important biological signals carried by a subset of cells that you cannot see in the traditional bulk RNA-seq data. And with this ability, you actually can do many cool things, like you can discover new cell types that you can not see in the bulk gene expression or bulk genomic data. And you can analyze cells' development trajectories, and you can analyze their spatial patterns. And you can also analyze cell-cell interactions in, for example, tumor microenvironment. And many of these things cannot be done using the conventional bulk technology. With these abilities, single-cell sequencing actually was named as the method of the year for 2013 by Nature Methods. And indeed, again, in 2020, Nature Method has named single-cell multi-modal omics as the method of the year for 2019. And in single-cell genomics, computational and statistical analysis actually play a very important role in processing the data, extracting information from the noisy data, and then turning the data into useful knowledge. And there are many analytical challenges in single-cell genomics, ranging from, say, data preprocessing, normalization, imputation, clustering, trajectory analysis, et cetera, et cetera. And today, I'm going to talk about one challenge related to a recent trend of single-cell genomic experiment. That is, the recent studies increasingly generate data from multiple samples with different phenotypes. And there, a question of interest often is, what are the features in the data that are associated with the phenotype, the sample phenotype? With multiple samples, now you're going to have more data. You're going to have more complex data structure. and you are going to have more information to digest. However, computational tools for analyzing multi-sample data have not been fully developed yet. And for today, I'm going to talk about two methods and tools we have developed for analyzing multi-sample data. One is called Trickle treat, and the other one is called Lamin. And Trickle treat is developed as a data exploration tool and to address the differential gene expression and cell type proportion analysis at cell type level. And Lamin is a tool developed for identifying differential patterns along pseudotemporal trajectories. Okay, so here is the Trickle tree. The Trickle tree stands for tree-based correlation screen and visualization for exploring phenotype-associated cell transcriptomic features and cell types. And this is a work with my PhD student Boyang Zhang, and also my former student Jason Ji, and also our collaborators from UPenn, Ze Yu and John Berry. And here is the bioRxiv link. If you are interested in details, you can look at the bioRxiv preprint. And if you want to use the software tool, you can go to this GitHub link, and you can actually download the software. And there is also a tutorial with very detailed instructions. So this slide shows an example of the type of data we are considering in Trickle tree analysis. So here, as you know, COVID has influenced our life for almost everybody. And in the past 2 years, actually many groups have applied single-cell RNA-seq to analyze COVID patients with different severity levels. And some of these studies actually generate hundreds of COVID single-cell RNA-seq samples. And there, a question of interest often is, what's the difference between a patient with different severity levels? Are there any cell types that are different between, say, mild patient and severe patient? And understanding this difference often can help give people some hint about the underlying mechanisms of disease severity progression. Right? And as another example, some people have applied single-cell genomic technologies to analyze cancer immunotherapy treated patients. And in one of our recent studies, indeed, we have used this approach to study non-small cell lung cancer. And there are some patients, we have patients treated with anti-PD-1, and we applied single-cell RNA-seq to analyze their tumor samples. And our question of interest in that study is, what are the molecular features associated with patients' response to the immunotherapy? As you can see, in both of these examples, you have single-cell RNA-seq data for multiple patient samples, and each patient has his or her own phenotype, and we are interested in identifying features associated with patient phenotype. So the question of interest, the first question people often ask is, which cell type actually is associated with the sample phenotype that you are interested in? So this question sounds quite simple, but indeed, when you actually try to answer this question, you find that practically answering this question is not that straightforward. It's actually very complicated. And there are many reasons for that. First, the phenotype cell type association can occur in multiple different ways. For example, for a given cell type, the sample phenotype may be associated with its abundance, say the cell type proportion. Or the phenotype could also be associated with the gene expression profile of that cell type. Or the phenotype can be associated with both cell type proportion and gene expression. And a priori, we don't even know which type of features, whether the cell type abundance or gene expression, is actually associated with the phenotype. And second, cell type actually can be defined at multiple resolution levels. For example, if you think about your immune cells, it contains, say, B cells and T cells, right? But among T cells, you can also have CD4 T cells, CD8 T cells. And even within CD8 T cells, you can have different subsets of cells with different effector phenotypes. So a priori, it is also known which cell type resolution actually provides the most strongest association with the phenotype. Therefore, it will further complicate your analysis. And also, a sample may have multiple phenotypic traits. In addition to, say, disease severity, you can have also other like characteristics of a sample, say whether the sample eventually recovered or whether the sample did not survive, and the age of the sample, et cetera, et cetera. And the association between phenotype and cell type can also be confounded by variables that you are not interested in, say the batch effect. For example, if you want to combine data from multiple studies, and there are obviously study-specific effect that you are not interested in and want to remove or account for. And because all of this indeed answer the question of which cell type is associated with phenotype is not easy. It's actually quite complex. You need to do many explorations, trying different feature types, whether it be cell type abundance or gene expression, trying at different cell type resolution levels, and trying to play with different phenotypic traits separately or jointly, and trying to adjust for different potential confounders, and adjust for age, adjust for sex, adjust for many other covariates, et cetera, right? To see whether your conclusions is robust, whether it's influenced by different factors or not. And this will amount to many combinations of analysis. And each combination of this analysis will require you to analyze hundreds of samples and tens of thousands of cells, which is not easy. So in that regard, exploratory analysis actually play a very important role in investigating the cell type phenotype association. And it's crucial that you can effectively handle these hundreds of thousands of different combinations of analysis to identify the major patterns. And also because you need to try these many combinations of analysis, you are going to get huge amount of information. Say for each cell type, you may identify hundreds of differential genes associated with the phenotype. Then when you try all cell types at different resolution levels, suddenly you're going to have tens of thousands of significant differential features. And how you're going to digest all these results is also not easy. And for that purpose, effective visualization of the information is also important. And as an example of good visualization tools that actually has long-lasting impact, just think about 10 or 20 years ago when people study DNA sequence motifs, right? So to visualize that information, people have developed this so-called sequence logo plot, which summarizes the motif information in a concise and informative way. And that actually has a huge impact, and many sort of papers today actually use this type of plot to visualize, to summarize information in the DNA sequence patterns. And similarly, in single-cell RNA-seq, when you consider this cell type phenotype association at multiple cell type resolution levels, how are you going to summarize all this information is not quite easy, and it requires an effective visualization tool, which currently I think is still lacking. So when you look at the existing software tools and methods, you can see none of these methods or tools actually can address all these challenges I mentioned in the previous slide. And there are some tools for bulk analysis that can handle multiple samples, but they cannot handle single cell level analysis. There are some tools for single cell gene expression analysis, but they do not handle, say, differential cell abundance, cell type abundance analysis. There are some recent tools that can handle cell type abundance differential analysis across multiple samples, but they don't address differential gene expression analysis. And also many of these tools do not explore cell type phenotype association at multiple resolution levels. And not all of these tools can provide an easy way to visualize the rich information you have in this data. So that motivated us to develop a trickle tree method, okay? And Trickle Tree actually is an open source R software package, so you can download it and try it yourself. And this slide summarizes the main pipeline of Trickle Tree. So first, we require you to provide some input data. So the input data for Trickle Tree consists of a gene expression matrix that contains expression values for each gene in each cell. And then you also need to provide a cell level metadata, okay? And then you also need to provide a sample level metadata. And cell level metadata basically tells you which sample are used to generate the cells, or like for each cell, where does the cell come from, right? Which sample does the cell come from? And the sample level metadata basically contains the sample phenotypes for each sample, what's the age, what's the sex, what's the disease severity level, et cetera, okay? And then with this input data, Trickle Tree will provide the whole pipeline to explore cell type phenotype association. The pipeline consists of multiple modules. In module 1, all the cells will be integrated and embedded into a low dimensional space. And by default, this is done using the Harmony pipeline, but users can also choose to use other methods like Surrey integration to integrate cells across samples. And after you integrate cells across samples, you can cluster cells into different clusters. So each cluster can be interpreted as a cell type. And then in module 2, users will give a name for each cell type based on some gene markers. You can do some differential gene analysis to identify genes that are, say, highly specific in a cluster. Right? So based on that, you can give each cluster a name. And then you can also specify a hierarchical structure of different cell clusters or different cell types. And for example, here we're considering a PBMC sample from the COVID patients. And in the PBMC, you have some myeloid cells, you have some lymphoid cells. Within the myeloid lineage, you have monocyte, et cetera. Within the lymphoid lineage, you have T cells, you have B cells, et cetera. And within T cells, you have like memory T cells, naive T cells, et cetera. Right? So you can, users can provide such a hierarchical structure of cell clusters based on their prior knowledge. And alternatively, you can also ask the algorithm, the Trickle Tree package, to automatically group the cell clusters using hierarchical clustering, and use the hierarchical clustering to automatically define this hierarchical structure. But either way, after this module, you are going to get a hierarchical structure of the cell types. And this tree structure will be used to guide our downstream analysis. And then in module 3, we are going to examine the association between sample phenotype and cell type proportion. And here, for each cell type or for each node in the tree, we're going to ask whether the proportion of that cell type in a sample, or the cell type abundance, is associated with that sample phenotype. And then in module 4, we are going to perform a global gene expression analysis. Here, we are asking for each node in the tree whether its gene expression profile is associated with the sample phenotype. And in module 5, we are going to go a step further and ask for each node or for each cell type, what genes actually associated with the phenotype in terms of gene expression. And finally, in module 6, we provide different ways to visualize the information for cell type proportion, global gene expression, or differentially expressed genes association with the phenotype. And starting from my next slide, I'm going to use the COVID single-cell RNA-seq data as an example to illustrate some of these functions. And in this data set, we have analyzed 371 COVID-19 samples consisting of about 700,000 cells. So after some data pre-processing and filtering out bad quality samples and cells, we have about 335 samples remaining, which contains about 600,000 cells. And then we first integrate them into this low dimensional space using our integration pipeline, and group them into clusters and specified the hierarchical structure of these clusters based on prior biological knowledge. And then we started to ask which cell type is associated with the severity of the COVID in terms of cell type abundance or cell type proportion. And then indeed, from this plot, UMAP plot, we have stratified patient into 4 groups: healthy donors, mild patient, moderate patient, and severe patient. And from this plot, you can already sort of see that the monocyte, which is this part of the plot, actually increases the abundance when the patient gets more and more severe. So that is reflected in our trickle tree analysis result, because you can see indeed this myeloid lineage, the monocyte and the classical monocyte actually is highly associated with disease phenotype. So to summarize this analysis we actually repeat this analysis for each cell type or each node in the tree and obtain the result. And then to summarize the result, we developed this so-called trickle tree plot. Okay, so this is a plot where you can show the association information for each tree node. So in this simple trickle tree plot, what I'm showing you here is the Pearson correlation between the cell type abundance and the disease severity. Okay, so the size of the balloon here, basically for each leaf node, you have a balloon to summarize the information. The size of a balloon characterizes the magnitude of the correlation. And the color of the balloon here indicates the direction, indicates whether the association is positive, which is purple, or negative, which is green. Okay, and the sort of grayness of these balloons indicate the significance levels. Like if it is not significant, you will have almost no color, very light color. If it is highly significant, you are going to have darker color. Okay, and as a result, you can see that from this analysis, the data indicate the monocyte, particularly the classical monocyte with high CD68 or intermediate CD68, is highly associated with, is positively associated with COVID severity. So more severe patients will have more these classical monocytes in their PBMC sample. And by contrast, lymphocyte, especially T cells, actually are negative correlated with COVID severity. This is consistent with previous report that severe patient often is associated with lymphopenia or lack of lymphocyte. And you can also see that indeed the negative association between lymphocyte or T cell with the severity occurs at this intermediate node, right? So comparing the leaf node and the intermediate node, the negative association is stronger in the intermediate node. Or in other words, if you want to have a clinical marker for the disease severity, you may consider use T cell as a whole as a marker. You ask whether the T cell abundance of T cell population as a whole you use that as a clinical marker for severity rather than sort of use each individual T cell subtype, right? So that illustrates sometimes this hierarchical or sort of multi-resolution analysis can help you identify the most appropriate resolution level to look at your data and study your sort of association in the data. And then next,
Dr. G,
yeah.
Hate to interrupt. I just have a quick question following the previous slide. I can include that. Yeah. Multiple questions around what is, do you know what is, I know it's from multi-study, but what is the tissue collected from this COVID patient? Is plasma?
These are PBMC samples.
Good. PBMC, got it.
Yeah. Okay, so the next question is whether the gene expression of each cell type is associated with disease severity. And for that question, we first consider one particular, say, cell type or node in the tree. And then we derive the pseudo-bulk gene expression sample for that node. And then we can project all patients into a low-dimensional space. Here is a principal component space using the pseudo-bulk gene expression profile of that cell type. And here each dot represents a patient, and the patients are color-coded based on their severity levels. Right? So in this particular example, you can see like patient somehow there are some association between the patient's locations in this embedding and disease severity. For example, here the purple ones are severe patients. You can see sort of severe patients are more likely to be located here, whereas mild and healthy patients are more likely to be located here. To characterize this association, we use canonical correlation analysis. Sort of we identify a direction in this space that most correlate with patient's phenotype. And as a result, we can say like for this particular cell type, the canonical correlation between disease severity and patient's gene expression is say 0.58. And we repeat this analysis for all tree node or for all cell clusters. And again, summarize the information here. And you can see that the cell types that are most associated with disease severity in terms of gene expression include, again, a myeloid and monocyte. And among the monocyte, classical monocyte and some non-classical monocyte actually show sort of high correlation with the disease severity. And interestingly, then B cells actually also show high correlation with disease severity in terms of gene expression, although each individual B cell subtype did not show very strong correlation. Okay, and within T cells, CD8 effector T cells actually show high correlation with disease severity in terms of gene expression. And in addition, proliferating lymphocyte NK natural killer cells and NKT cells also show strong correlation. So this is sort of an illustration of the global gene expression analysis, asking whether the gene expression of each cell type is associated with phenotype. And then if you want to know which genes are associated with the phenotype, you can also sort of do a sort of more detailed analysis. Here within each cell type, we run a lemma analysis and ask which genes actually show association with the phenotype within that cell type. So basically for each cell type, again, we convert the data into a pseudo-bulk sample for each patient sample. And then you have multiple patients and each patient has a phenotype. So you can imagine that sort of is similar to a traditional bulk gene expression data set. You can then ask what are the differential genes, right? And then this trickle tree plot then summarizes the number of differential genes we have found for each cell type. And based on this result, you can see proliferating lymphocyte actually shows the highest number of differential genes. And the other like cell types that are highly associated with phenotype include, again, classical monocyte and some non-classical monocyte. Both of these cell populations actually have many genes that are differentially expressed between different severity levels and also CD8 effector T cells. So many of these analyses actually are quite consistent with the global gene expression analysis we have, okay? So in this way, with a new data set, you can sort of quickly take a look of the information in the data and understand which cell types actually are associated with the phenotype of interest and whether the association occurs at the cell type abundance level or whether the association occurs at the global gene expression level, and also how many genes actually are differentially expressed related to a phenotype of interest. And all this has been summarized using a trickle tree plot. Actually, we have developed a special trickle tree plot function that supports sort of a very flexible, customized visualization of information from multiple resolution levels. And in my previous example here, we only sort of use disease severity as one sort of phenotype or outcome. But indeed, you can sort of display multiple analysis within the same trickle tree plot. For example, we have collected COVID-19 data from multiple different studies. SS1, SS2 are two patient cohorts from one study, and SU represents another study. And if you want to examine whether your results actually are reproducible or consistent across different patient populations or across different studies, you actually can run the analysis for each study and display their result in the same trickle tree plot. And for example, if we examine the cell type proportion analysis, we can see that, so for example, the proliferating lymphocyte is consistently positively associated with the disease severity in terms of the cell type proportion, whereas some of these other cell types do not necessarily sort of show consistent association patterns. In other words, their sort of direction of association or their significance of association varies from study to study. And similarly, you can sort of examine the consistency at a gene expression level or like a number of differentially expressed gene level. And in the trickle tree plot, we give users sort of, options to specify the way to visualize the tree. For example, you can use these straight lines, or you can use these curved patterns. And then you can show your information using the balloon plot or heat map or bar plot, etc. You can sort of specify which nodes to highlight using the colors. And you can also specify how to display the result, whether you want to use color to indicate the significance level, or whether you want to use color to indicate the direction of association, etc., etc. So there are many options users can use to decorate this trickle tree plot and to present their information, okay? And then we also, of course, provide a function for covariate adjustment. For example, if you want to integrate these three studies, because there are some inconsistency, suggesting there's some batch effect. So if you look at these three studies, and here each dot represents a patient sample, you can see like a patient sample from different studies actually separated, suggesting a strong batch effect. If you want to combine data from the three studies, you want to remove the batch effect first. So one way to do that is to incorporate the batch as a covariate, and then you adjust for that covariate, and then you do the downstream analysis. And hopefully that can help you get more samples to increase the power of your analysis, but still allow you to avoid the negative impact of batch effect. So this shows an example. So after you treat batch as a covariate, adjust batch effect, the samples are now mixed together, suggesting batch effect has been removed. And then you can again do the trickle tree analysis. And here is just an example of the trickle tree analysis. You can see like before batch effect correction and after batch effect correction, some of the results actually have been changed from, say, non-significant to significant, for example, for the CD8 effector T cells, okay? So this is just a sampling of the functions we have provided in trickle tree. There are many more functions, and unfortunately, in the interest of time, I cannot go through all of them. But if you are interested in those functions, you are certainly very welcome to read our trickle tree manuscript and also try our trickle tree software. So to summarize, the trickle tree is a software tool that supports multi-resolution phenotype cell type association analysis with multiple single-cell RNA-seq samples. And particularly, we have developed trickle tree plot as a new visualization tool to support your multi-resolution data exploration. And we hope with this tool, you actually will find it easier to explore the rich information in the multi-sample single-cell RNA-seq data. All right.
Dr. Ji, I hate to interrupt, but because we have a very enthusiastic audience, we have several questions, I think, pivoting around how we integrate multiple data sets. I think I think you addressed some part of it. You know, if we have the freedom to do batch correction, but if I'm getting this correctly, so upon integrating multiple data sets that we actually may or may not correct for batch effects, do we perform any, for example, harmony or several other data, integration methods, to combine the data. But if I'm understanding this correctly, after doing the analysis, we also can actually visualize the significant result back into each individual study. Is that correct? But we sort of want to do some data integration together in the first place.
Yeah, so the data integration actually occurs at multiple levels, right? So if you go back to this slide, the first data integration step is integrate cells from all the samples. Here, if you have multiple studies, you want to integrate samples from all studies together. So the purpose of this integration step is just to match cell types so that T cells from one sample is matched with T cells from another sample, regardless of which study generated that sample, and the B cells is aligned to B cells. So that later in your analysis, you know that you are comparing apples to apples and oranges to oranges. Right? You are comparing T cells across different patients. You are comparing B cells across different patients. And you are not comparing B cells from patient A with T cells from patient B. So that's the purpose for this integration. But that's the once you are done with this, and this integration is no longer used, and when you start to do this differential gene expression analysis, you have to go back to the original gene expression values, and those expression values are not batch corrected. Okay, so the reason is so in this integration, some authors also provide sort of corrected gene expression values. So when they integrate different samples, they basically remove the sample differences. But there, the sample differences can contain true biological differences, and it can also contain batch effect. If you remove all the sample differences, you actually remove all the true biological differences. So that's not what we want. Right? So therefore, when you do this differential gene expression analysis, you have to go back to the original gene expression values. And you only use this integration to specify which cell type it is. And then once you go back to the original gene expression values, you are sort of going back to the batch effect correction problem as you have in the traditional bulk RNA-seq data.
Got it. All right, let's move on. Yeah. Okay, thank you. There's several questions addressed, I believe. Okay, let's keep going. Thank you.
Okay, so then next is Lamin. I'm going to quickly go through Lamin given the time remaining. So Lamin is a method to analyze differential trajectories. So first, for those who may not be familiar with trajectory analysis, it's a way to analyze a continuous biological process using single-cell genomic data. So it's pioneered by Cole Trapnell's Nature Biotech paper on monocle. So in their paper, what they're saying is you actually can project cells from a sample into a low-dimensional space, and here each node represents a cell. And then you can construct a minimal spanning tree to link all these cells together. Here, if two cells are close to each other, that means their gene expression profiles are quite similar, right? So you can define a distance between two cells accordingly. And the minimal spanning tree basically is a spanning tree that connects all the nodes, and so that the total edge length of the tree is minimized. And then they show that if you have this minimal spanning tree, often this tree structure reflects the underlying sort of biological process. For example, if you move from one end of the tree to the other end, you sort of can capture the differentiation process. Like one end of the tree represents undifferentiated cells and the other end represents differentiated cells. And then so that you can sort of move from this end to that end to sort of recapture the process that cells gradually differentiate from a undifferentiated stem cell to a terminal differentiated cell, something like that. And then as a tree, it has branches, right? So it turns out different branches can represent different lineages of cells. Like some cells are ectoderm, some cells are corresponding to mesoderm, et cetera, et cetera. So in this way, you can order cells along this trajectory to turn this whole data set into a pseudo time course data set. So for each cell, you now have a pseudo time, right? So you can arrange cells based on their pseudo time, and then you get a pseudo time course. And for each gene, you can ask how that gene changes its expression level along this pseudo time. And then when you repeat this for all genes, you get these pseudo temporal gene expression programs for all genes. And you can ask which pathways are like turned off or turned on as cells differentiate from stem cell status to a fully differentiated status. Okay, so that's the basic concept of pseudo-time analysis. And indeed, after that pioneering work, and hundreds of actually methods have been developed for pseudo-time analysis, and it's actually also reviewed and benchmarked by this paper. And however, when you look at all these existing methods, you realize that actually very few of them, or almost no, I probably should say to the best of our knowledge, right, when we start to work on that problem, no method actually tackled the analysis with multiple samples, right? Even though such studies, as I said before, become increasingly common. So that motivated us to develop our Lamin method. And the first, what would be the problem if you don't develop this new method, if you just use existing method? Okay, so existing method mainly considers the problem of constructing a pseudo-temporal trajectories using one sample. Say they assume you have one biological sample, and then that sample contains many cells, and how to construct the pseudo-temporal trajectories using all cells, right? So that's what most current methods do. So if you have data from multiple samples, and if you want to use existing method, the only way you can do is you pull cells from all the samples and treat them as if they are from one single sample, right? But we know if you do that, you probably ignored the important sample level variability structure, and that will give you very misleading result, because as a statistician, and many of you are statisticians, and also as data scientists, you know that cells from the same sample actually may not be totally independent. When you treat all cells from different samples as if they were from one sample, you are basically assuming that all these cells are sort of exchangeable. They are sort of different samples are sort of exchangeable, and all cells are independent. And then that will sort of artificially inflate your sample size, and your true sample size actually is much smaller than that. And as a result, you are going to have like too optimistic, say, p-values or statistical significance. Okay, to illustrate that, you can look at this example. In this example, we have, say, 8 samples, 4 males, 4 females, and each curve here represents a sample level curve along the pseudo-time. X-axis is pseudo-time, y-axis is expression of a gene. So you can see the 4 male samples have huge variability across different people, and the 4 female samples also have huge variability. And when you look at these two groups, the 4:4 comparison, you can see like there's no obvious difference between males and the females. Actually, when you do statistical test, there's no significant difference between the two groups. However, if you pull cells from all samples together and just feed a male-specific curve, which is the, say, blue curve here, and feed a female-specific curve, which is the yellow curve here, using existing method, say, Tde-seq is one of such method, you are going to claim that these two curves are highly significantly different because there are so many cells supporting the difference between this blue curve and yellow curve. As a result, you are going to report this gene as a differential gene between male and female, while indeed there is no significant difference between these two groups. As a result, you are going to get many, many false positives. So that's the problem with existing method if you don't consider the multiple samples. But that's not the only problem. There are also many other problems. For example, few of the existing methods actually considers the variability of the tree topology. Right? So if you consider the trajectory of the tree, so it has some kind of a branching structure. If you do this for different samples may give you slightly different structure. Like for this sample, you may have a small branch here, whereas in the other samples, you don't have that branch, right? And few of the existing method actually considers this variability in terms of the tree structure, okay? And also this uncertainty in tree structure can also influence your downstream analysis of the differential gene expression. Right. So there's some initial effort, like by Jessica Lee's group from UCLA, they tried to incorporate the uncertainty of the pseudo-time in the differential gene expression analysis. But even for that method, the author did not consider multiple samples, they only consider sort of one sample. Okay, and also changes across patients may occur in either gene expression or cell type abundance, just like I talked about in the Trickle tree work. And here is similar, but not all method actually handles both types of differences when they perform the trajectory analysis. So that motivated us to develop the Lamin, which is a statistical framework for differential pseudotime analysis with multiple single-cell RNA-seq samples. And then this figure summarizes the workflow of Lamin. Again, here you have multiple patient samples, and you have a covariate or phenotype attached to each patient sample. And the first step is we are going to integrate cells from all patients together by embedding them into a low-dimensional space. And second step is we're going to construct a cluster-based minimal spanning tree, meaning that we first cluster cells into different clusters and then use those clusters as a node of the tree to construct a minimal spanning tree. That will give you a backbone of the pseudotime structure. And then once you have this backbone, you specify a starting point of your pseudotime, and then you are going to enumerate all the paths of the pseudotime. For example, here, if the starting point is cluster 8, then you have 3 trajectories: 8, 7, 4, 1, 8, 7, 4, 3, 2, 8, 7, 4, 5, 6. You enumerate all the trajectories, and then subsequently we are going to break it into different branches. Here you have 4 branches: 8, 7, 4 as the first branch, and then 4, 5, 6 as the second branch, 4, 3, 2 as a third branch, and 4, 1 as the fourth branch. And then we are going to evaluate how reliable each of these branch is by using some kind of bootstrap procedure. We're going to bootstrap cells from each sample and repeat this procedure and see whether we can sort of confidently reconstruct each of these branch. And then we're going to compute a detection rate for each branch. For example, here branch 1 will have low detection rate, meaning that it's not that reliable, where 874 is a branch with high detection rate, meaning that it's very reliable. Conditional that on that you are confident that a branch exists, then we are going to test whether the cell abundance for that branch is different between different sample groups. And remember, for each sample, we have a sample phenotype, we have covariate, right? So we can test whether the cell type proportion in that branch is associated with that sample phenotype. That is our second module, which is the differential topology analysis. Because if say you are comparing males and females, if males have this branch, but females does not have this branch, then that means for this branch, the females will have 0 proportion. The proportion of cells in that branch is almost close to 0, whereas for males, you have a relatively larger proportion. Then you are going to see the difference in terms of cell proportion for that branch. So that indicates sort of the topology between the two groups, sample groups are different. So that's the first type of differential analysis we do, which is we test differential topology. And then the second type of differential analysis we do is we test whether gene expression actually are different along the pseudotime and across different patient groups. So here we have 2 types of differential test. One is called Tde test. In this test, we ask whether a gene changes its expression along the pseudotime, considering the sample level variability. So this test is similar to the existing methods like Monaco or others, which also want to test whether genes change along the pseudotime. The difference is that the existing method does not consider sample level variability, whereas Lamin considers sample level variability. And as a result, we sort of eliminate a lot of false positives and can give you more sensitive result. And the second type of test is a new test, which is Tde test. Here we want to test whether different sample groups have different temporal gene expression patterns. For example, for one gene, you have gene expression patterns for one group of patients indicated in the green. You have gene expression pattern in another group of patients. Whether these 2 groups of patients actually show different patterns, we call it a differential gene associated with sample covariate X, and therefore Tde test. Okay. And similarly, we also test a cell type abundance. And there are 2 types of cell type abundance tests. First is TCD test, which tests whether cell type abundance changes along pseudotime by considering variability across samples. And second type is Tde test, which tests whether the cell type abundance patterns along pseudotime are different between different sample groups. Okay, so these are sort of the basic pipeline of Lamin. I'm not going to talk about the details, but essentially we use a mixed effect model to consider multi-levels of variability, including sample level variability, cell level variability, and also we use splines to model the curves, and then we develop an EM algorithm to fit this model. And I think I probably want to skip this part. This is just an illustration of the method. Instead, I'm going to directly go to some benchmark result to hopefully convince you that Lamin actually provides some unique advantage compared to the existing method. And first, as I said, by considering sample level variability, we actually can eliminate a lot of false positives. Indeed, in this regard, you can do a null simulation. Say you have a data set, right, with multiple samples. You can randomly split them into two, and then you don't expect any difference between sort of, the samples between the two groups because that are randomly partitioned, right? Whereas if you use existing method to analyze this type of null simulation data, what you see is often the existing method report thousands of significant differential genes, which are all false positives. And the reason that the report is false positive is, as I said, they don't consider sample level variability. Whereas if you use Lamin, you basically get zero or close to zero false positives. So that says accounting for sample level variability in multi-sample studies is really important. Otherwise, you probably will spend all your money or like time to validating those false positives. And then to evaluate sort of sensitivity, we did some benchmark simulation where we spec in like computation specking some true signals to the null data set we have. And then we did some benchmark. And as a result, you can see that in terms of the ROC curve, the receiver operating curve, the area under the curve, Lamin actually provides the best performance. Okay, the red curve is Lamin, whereas the other methods has lower sensitivity, specificity trade-off. And also in terms of comparing the true FDR and the reported FDR, you can see Lamin usually controls FDR. Here, what I show here is for different simulation settings, what's the difference between real FDR and true FDR. If it is negative, it's better, it's good, means like true FDR is smaller than what you reported. And Lamin always like is negative, whereas this other method, many of them actually provides sort of too optimistic significant conclusions. And then we have applied this again to COVID-19 single-cell RNA-seq data set. And here we are studying a T-cell activation procedure. So we collected naive T cells and CD8 T cells and construct a pseudo-temporal trajectory. Here we only have one trajectory with no branches. And by detecting TDE genes differential across the pseudotime, we can see that this pseudotime actually reflects the T-cell activation. For example, naive T-cell markers, memory T-cell markers are enriched at the beginning of the pseudotime, whereas terminal differentiated T-cell markers or like effector T-cell activation like markers are activated at the end of the pseudotime. So we are sure that this is a T-cell activation process. When you look at the cell type abundance along the pseudotime, you can see that here we focus on the comparison of mild COVID patient and moderate COVID patient, because this is the transition stage that most differences occurs. And you can see that moderate patients actually have higher proportion of cells at the end of the pseudotime, suggesting that these patients have more like effector or terminal differentiation cells, right, compared to mild patient. And when you do like differential gene expression analysis between these two types of patient, we actually detect the hundreds of differential genes grouped into different patterns. And then, for example, just took one example, and here the cluster 1 is a set of genes where the moderate patient, the origin here, showed lower expression compared to mild patient. And the expression level, the difference between the two actually increases as you progress along the pseudotime. And when you look at the genes in those in this cluster, you can see that these genes actually correspond to like T-cell effector functions, and this suggests that so as the pseudo-time T cell activation process progresses, and the mild patients shows a more robust effector function compared to moderate patients. And similarly, you can derive interpretation for these other sort of categories of differential genes. And then we sort of also did another benchmark using this real data to see whether Lamin actually is advantageous compared to the other method. And for this test, we split samples into 2 groups. And then we perform analysis for each group separately. And we ask how consistent or how reproducible are the results we obtained from these 2 groups. And the good method should give you more reproducible result when you use different samples to study the same phenomena. Right? So we compare the overlap between the differential genes reported from these 2 partitions of data by each method. You can see Lamin actually shows the highest reproducibility across different showed the highest reproducibility between the 2 partitions of the data, whereas the other method showed much lower reproducibility. So this suggests actually by accounting for multi-sample variability, we actually can achieve a better performance. Okay, so with that, I'm going to conclude this section. So to summarize, Lamin provides a new tool for pseudo-temporal trajectory analysis with multi-sample single-cell RNA-seq data. It actually provides a comprehensive solution for topology uncertainty assessment for detecting differential topology, differential gene expression, as well as differential cell abundance, both along the pseudo-time and across sample covariates. And the key take-home message is by accounting for sample level variability, Lamin properly controls false discovery rate and offers higher sensitivity in these analyses. And with that, I'm going to conclude my talk today. I've introduced Trickle tree and Lamin for 2 types of differential analysis, and there could be many future extensions like extending into other single-cell data types and multi-omic analysis, et cetera, And I want to thank all the authors who contribute to this work, as well as funding and computing resource provided by NIH and also by Hopkins High Performance Computing Clusters. Okay, so I'm done with my talk.
All right.
Thank you for your attention.
Thank you, Dr. G. And I want to thank the audience for keeping on the channels, and we have almost 150 people join today. This is a solid, informative slide deck, and thank you, Dr. G, for coming giving this talk. So I know we're running over time, so I'm just going to very quickly sort of combine several questions. I think they're all pivoting around the trick or treat method. I think 2 questions I want to ask on behalf of the audience to Dr. G. First of all, it's also one of my questions is that how do you think that trick or treat can handle data set that are small sample size? For example, like we're looking at several hundreds of thousand cells from multiple COVID studies. But what if like sometimes when we have a study design that's SmartSeq, that we don't have a lot of cells, or study design small, how robust do you think trick or treat can still perform in identifying all of the lineages in the trees? And second question, I think a lot of people are asking is, how did you define your cell types? Is it a manual process, or could the a package actually aid in, provide some input to the cell type identification?
Okay, so both of these are good, very good questions. So first, how to define cell type. So for the cell type, you actually there actually are many softwares you can use, right? So within the Trickle tree, what you do is you can detect differential genes, which actually we sort of used a function in Surat to allow you to define cell type specific marker genes. When with those marker genes, the next step is you either ask your expert biological collaborator to annotate the cell type based on those marker genes. Say if you have a marker gene for T cell, CD8 T cells, you see that you annotate using as CD8 T cells, things like that. Alternatively, you can also use many existing tools and to annotate cell types automatically, although our experience that automatic annotation often is not does not satisfy our collaborators' prior knowledge. So they often come back and intervene our annotation. So therefore, we sort of provide users this option of defining your own cell types. You basically input your own cell type labels based on sort of your own annotation or based on like combining your knowledge with some automatic annotation. Okay, so that's provided as an option. And then regarding the sample size and robustness, there are two sort of sample size in some sense. One is the number of cells, one is the number of samples, right? So I think for when you talk about say like number of cells, if you don't have that many cells, that's okay as long as you have many samples. You can still do this analysis because if you have like fewer cells, you can still do the clustering, right? Because we can do clustering as multiple resolution levels. When you go to the higher resolution levels, each cluster will still have many cells, right? So maybe to the leaf node, if you have too many clusters, each leaf node may only have like 1 or 2 cells, then the analysis may not be that robust. But since you have this multi-level analysis, you actually can look at the intermediate node, and there the analysis would be more robust. And then even for the leaf node, when you have small number of cells, when the measurement for that leaf node is not robust, that's also okay, because that means the variability across samples you see will be very big. And as a result, when you do the statistical analysis to test whether, say, gene expression of that cell type is associated with phenotype, just due to the high variability for that node, you probably won't be able to see statistical association with the phenotype. So that small will be automatically filled out, right? And then in terms of the sample, patient sample size, like how many patient samples you have, of course, the more samples you have, the more robust analysis will be, right? So if you only have a few number of patient samples, like 3 or 4 samples, then some analysis can be done, some cannot. So like for the differential gene expression analysis, you can still do it with only like 2 or 3 patients per group. So that's that was done like traditionally in Lima and in say Tde or in other bulk RNA-seq analysis. So you can still do it here, right? And then for like cell type proportion analysis, if you examine simple cell type proportions, like what's the proportion of this cell type, for those simple Pearson correlation analysis, like with 6 or 7 samples, you can still do that correlation analysis relatively reliably in a relatively reliable way, right? But you just have a higher uncertainty level, but you can still run that, right? And for this global gene expression analysis, similarly, you can still do it. It's just you are going to have higher uncertainty. Indeed, in one of our like immunotherapy study, there we only have like 10 to 20 patients. We can still do this analysis, and the consequences we can see there is some separation between different patient categories, but the statistical test, the p-value may not be significant. Say you got a p-value of like equal to 0.1 or 0.2, right? Which does not pass your significant category because you don't have enough power. But by eye, you can see the separation of different patient categories in the plot. So suggesting there probably are some biological difference. It's just your study is not powered enough to claim that you can significantly identify those differences, suggesting you may want to expand your study to include more samples. So as an exploration tool, you can still use it. Okay, so hope that answers your question.
Thank you, Dr. Ji. And I just want to remind everybody that a lot of questions are very good. They're a little bit technical. I'm going to have them summarized and return back to Dr. Ji, and hopefully we can have them all addressed and posted to our YouTube channel that we wouldn't be able to address a day. Pay attention to the comment region of the YouTube channel when you want to revisit this talk. So Dr. Ji, I have one last question. I think it's going to be very straightforward and quick. Is for Lamin and Trick or Treat, when we're using the 2 packages dealing with large data set like the COVID with hundreds of samples, hundreds of thousands of cells, excluding manual part, how computational, what is the general computational time that these are going to acquire? Is it going to rely on the high computing service, or you can run it locally on a laptop with some multi-core CPU or something like that? How many hours or?
That's a really great question. And then you actually can find this computational requirement in our preprint. And then, but briefly, for like Trick or Treat, the main computational resource heavy part is this integration part, which relies on harmony. once you have done harmony, all this remaining part actually is not that resource intensive, and you can deal with handle it very quickly using sort of a not that powered computer. It's okay. And for the Lamin analysis, the old version that in the preprint, the version we showed in the preprint is a relatively slow version. For that, you need multi-core and a lot of memory to run like the hundreds of COVID samples, for example. But we are sort of currently revising our manuscript based on reviewers' comments, obviously, when we are trying to reduce the computational burden. So in our newest version, which has not been released yet, but we expect to release it very soon with our revision submission. And then that actually becomes also quite efficient because we did a lot of optimization in terms of how to handle like memories efficiently and data storage efficiently. So that again, the most time sort of consuming step is that integration step, as well as some of the sort of curve fitting step. But in general, it's sort of much more efficient and it's doable definitely for, I think, for most of the people, I guess. If you have a
sounds good.
Yeah.
All right. Thank you very much, Dr. G. I think we're running significantly over time. And I want to thank you for coming, and I want to thank many thanks to the audience for staying on the channel. Dr. Z, we'll always welcome you back. Maybe later in the year or next year. Always feel free to come back whenever you have new publications. Say, hey, got this new thing that's popping up. Want to draw some attention. This is a great opportunity for us to learn. So I wish you a happy new year, and thank everybody, and stay tuned for our next seminar next month. Thank you very much.
Sure. Thank you so much.
Yeah. Okay. Everybody have a good one. Bye-bye.
Bye.